So I'm Paul Jaeger, and I will talk about the current challenges and opportunities towards transforming healthcare with general purpose AI. And I would like to start by convincing you that we really need AI to address the current and future challenges in healthcare. So I just learned from Sarina that this consortium is all about visual intelligence, but not necessarily healthcare. So I'm going to give this little healthcare introduction here. So I argue that we face an imminent data explosion in healthcare with around 30% of the world's data volume generated in this domain and a growth rate of around 36%, which far exceeds other industries. And two factors driving this growth are aging societies, as well as the cost reduction in data acquisition, such as genomic sequencing and imaging protocols. It's also known that processing and analyzing this flood of complex data continues to exceed human capabilities. So one remarkable number in this context is that already today between 30 and 50% of physicians are thought to be affected by burnout. And now, given my background in image analysis and your focus on images, my talk will have a focus on medical images as a data modality. So I'd like to highlight the important role of images in this context. So they make up around 90% of this data. And also a study in Lancet Oncology found that scaling imaging to the global demands could save nearly 10 million cancer deaths in the next years. So I hope these numbers made it clear that we need an AI-driven transformation of healthcare. So where are we today? Let's have a look at the current status of clinical AI integration. So the current approach is basically to train and develop a specialist system for each new task. Meaning for each new task, we have to annotate a large number of training cases and then meticulously design and configure a model for optimal performance on this task, right? So by October last year, there were 692 AI devices that had been FDA approved, and most of them focus only on one or two narrow tasks. And the fact that 77% of these are in radiology, again, highlights the pivotal role of images in healthcare. So now we might think, well, this number 692 doesn't sound so bad, right? So the question is, will this current approach of specialized models scale to drive the required healthcare transformation? And my answer to this question is a clear no. So let's look at some challenges in the field that I think will prevent this approach from scaling. So for one, we face an extreme diversity of clinical diseases as well as data shifts in this domain. To give an example, in radiology alone, the catalog of disorders sums up to around 20,000. And around half of these are in the so-called long tail of rare diseases, meaning that the collection of sufficient training data for each of these will be almost impossible. I remember I had a colleague during my PhD, Sarina, you might remember Andre Klein, who was tasked to develop a deep learning system for detection of rare bone cancer. And even after 3 years and multiple clinical collaborations, his data set consisted of 7 patients. So this gives you some perspective on the scale that we're talking here. Okay, but let's assume we were able to collect the training data for each of these disorders. We would still be facing the second challenge of diverse data shifts in the medical domain, meaning when applying AI systems in the wild, the data may shift due to varying image devices, population characteristics such as age, race, or sex, or subtle semantic variations such as a new pathology subvariant of a tumor. And one remarkable example for what happens when we ignore such data variations is given in a meta-study that analyzed 62 proposed systems for COVID detection based on chest X-ray. And the study found that none of these 62 systems was of potential clinical use due to a high risk of bias. And one of the most prevalent pitfalls here causing a bias was that numerous studies did not control for patient age in their training data. So they had in the training set grown-up patients with COVID infection, and the healthy patients were basically kids. So the model only learned to distinguish between grown-ups and kids, right? And this was pure shortcut learning and not the underlying pattern of COVID detection that was intended. Okay, but even if we were able to collect sufficient training data in all its diversity, so we're still facing the bottleneck of missing ground truth for AI training. The problem here is that for most data cases, the true biological information is unknown because it would require biopsy or other invasive procedures. So most training on medical images relies on human references, annotations based on the images. And this process is especially time consuming in the medical domain and requires expert knowledge. So for instance, annotating a brain tumor in a 3D image, like you would need it for subsequent radiation therapy, may take up to 1 hour per patient. And also these image-based annotations are often ambiguous with a high intra and inter-rater variability that leads to noisy training. In this example here, a study found that the pairwise overlap between raters of liver tumors is only 80%, which makes a difference for a subsequent radiation procedure, right? Another major challenge is the human factor of clinical AI integration. So since the liability of life-critical decisions will remain with the human, the question is, how can AI really effectively and reliably support the human? Or how can human and machine form a symbiotic system for enhanced clinical decisions? An example for this bottleneck is the fact that most of the nearly 700 FDA-approved devices that we saw are currently not used. So on the top left plot, you see that current usage is dominated by only 3 or 4 systems. And this highlights that only because a system works does not guarantee its clinical benefit and its adoption in practice. Another challenge in this context is automation bias, meaning that human decisions may be negatively affected by over-reliance on the AI prediction. So a recent study found that especially inexperienced radiologists are prone to this bias. And the final challenge that I would like to highlight is a bit of a meta topic and describes a general disconnection between research and clinical application. So this gap is often referred to as the AI chasm or the translation gap. And the problem is that current research in the domain is somewhat inefficient, meaning that the majority of the resources we invest as researchers have no real clinical impact. The COVID study from before was an impressive example also for this gap, right? And now we can distill each of these 4 challenges into 4 main research questions that need to be addressed in order to achieve clinical integration at scale. And now in this talk, I would like to show you how I address each of these questions in my research. So we'll highlight some publications that I did in the last 3 years. And then finally, I will discuss how foundation models may help to address the data challenges. And I will show some of my ongoing efforts in this area. So let's start with the first question. To cope with the extreme data diversity that we saw, how can we build models that really generalize across such data sets and variations to allow for broader application? And I would like to show you how we tackle this problem, specifically the current need for manual model tuning on each new task. And I know you're all sort of familiar with this problem of hyperparameter tuning and so on, but I would like to make the point that it poses an even bigger issue in the medical domain because 1, the complexity and diversity of data requires more tuning. 2, the training times are rather long, leading to slow iteration cycles. And 3, that medical images, especially in 3D radiology, feature a very low signal to noise ratio. You're often looking for needles in haystacks, so to say, like a small tumor in a large 3D volume. So training is typically patch-based to oversample informative regions and also to fit large volumes into GPU memory. And this patch-based approach adds an extra layer of engineering and complexity. So the fact that this process is time-consuming and requires expert knowledge means that it bars access to AI methods for many users like clinicians. And it also has adverse effects on the research landscapes because it leads to low-quality baselines and so unreliable claims of methodological progress and so on. Now, to address this in 2021, we presented NNUNet, which is a generalist AI method in the sense that it adapts itself to new segmentation tasks and data sets. So since adaptation includes the entire processing pipeline here, this means that users can really use NNUNet out of the box on their problem without requiring expert knowledge. And here's how it works. So in contrast to AutoML approaches, which is sort of the related task, there all hyperparameters would have to be learned from scratch on each new task based on the data, right? But we follow a pragmatic approach that only takes 1 or 2 minutes to compute and requires no extra training data or large-scale hardware like AutoML would. So in the first step, we want to take as much complexity out of the process as possible and identify these fixed parameters, meaning all design parameters that generalize across tasks and need no adaptation. And you can see actually quite a few important parameters we found that they don't need adaptation, right? Like the learning rate and so on. And we also identified a joint configuration for these fixed parameters that ensures optimal performance. In the second step, we develop a fingerprinting process where characteristics of the data set are automatically captured and fed to a system of heuristic rules. So those are basically if-else statements that model the interdependencies of parameters. So we call this group the rule-based parameters. So it's like if the patch base is set to be like this, then a batch size of X fits into memory and so on. And then it can also iterate around until it finds a nice configuration. And with this configuration, up to 3 unit architectures are trained, featuring 2D, 3D, and a cascade unit. And in the final step, only 2 remaining parameters are determined empirically based on the training outcome. And importantly, none of these internal processes affect the user, who can really use the system as an out-of-the-box segmentation method. Now, this method works remarkably well. So here you see 53 segmentation tasks, all in the form of leaderboards from international competitions. And our unit is the red dot, and each blue dot is a competing team. And we can see that NNUnit takes the first place in 33 out of 53 tasks. And importantly, in the remaining 20 tasks, NNUnit is on par with or close to the winning submission. So it shows top performance in a robust manner. And to get you an idea of how surprising these results are, you would not expect a decathlon athlete at Olympic Games to outperform the specialized athletes in their respective disciplines, right? But this is kind of what happens here. So NNUnit is applied without any extra tuning to the task and competition or any manual intervention, and it competes against these specialized and tuned solutions. And the credits for this engineering effort here go to my friend Fabian Isensee, who was the technical lead on this project. And since we make claims about our auto-configuration generalizing to new data sets, it's important that we perform the development of our method on a restricted pool of development data sets that's highlighted in yellow here. And then we fix the whole setting and apply the fixed method to new application data sets. This is the only way we can test if it generalizes, right? So that's sort of like a clean train test split, but on a meta data set level. But the results you see here are all still based on unseen online test splits within each data set. Right, so to show you some qualitative results, I hope this gets you an idea of how diverse the application scenarios are. They range across different modalities, organs, tumors, lesions, and cellular structures in 2D as well as 3D. And this had quite an impact on the field as measured by citation and so on. But I think the impact on the field is actually nicely measured by its widespread adoption. So a study found that NNUnit was used by the winning teams in 9 out of 10 segmentation challenges that were hosted at MICCAI in the same year. Right. Okay, so one question might be now, does this only work for segmentation problems, this principle? To answer this, next we applied the same auto-configuration principle to 3D medical object detection. And this method is called NN detection. I know it's not very original, and this was published at MICCAI later that year. And the challenge here was to cope with the increased engineering components that object detectors have compared to segmentation models. And this required to come up with new heuristic rules, such as for the systematic configuration of anchor boxes and so on. And we were able to show the benefits of these methods again on a large and diverse collection of data sets. Okay, now let's take a step back and look at the concept behind these methods. So given a diverse set of custom problems and data sets, we systematically extract the relevant characteristics of each problem to form a task fingerprint. So a generalizing solution operating, we have then a generalizing solution that operates in this fingerprinting space based on heuristic rules. And these rules are executed really fast to generate an immediate customized solution. Right? This sounds a little bit abstract, but I hope with the NNUnit example in mind, you know what I mean here with these sort of abstract descriptions. The point is that we can apply this recipe to many more problems. So we have seen it in action now for configuration of segmentation, object detection, but we also applied it to other parts along the image processing pipeline, so including data annotation or method validation. So it's really a generalizing concept that addresses the extreme diversity of data and tasks in the biomedical domain. So let's move to the second critical question that I motivated before. How can we overcome the annotation bottleneck? Looking at it again at NNUnit, we may have solved the problem of configuration, but the bottleneck remains that methods have to be trained from scratch on each new data set, requiring substantial annotation training data. Now, Fabian, again, the guy who engineered and implemented NNUnit, he now runs a service lab where they basically guide researchers from different disciplines to solve their image analysis problem. So people often approach him with their data set and want to apply NNUnit. but they have no annotated training data. So interestingly, by trying to circumvent this problem, Fabian and his team converged to a working mode that's sort of a human active learning loop. So where first NN unit is trained on a small starting budget and applied to the entire data pool. And then a machine learning expert goes through all the predictions and manually flags errors and uncertainties and gives them to the customer for a second annotation round and so on. Now, this, of course, saves annotation time because only the most informative samples are selected for annotation. However, the process shifts the time-intensive labor to the query stage, where informative samples are identified by sifting through the entire dataset. And you can imagine that this approach does not scale to large datasets. So this bottleneck motivated me and my team to develop a true active learning method around NN unit, which, again, not very originally, we call NN active. And the goal is to automatically identify the most informative samples based on uncertainty and clustering. And now, crucially, we are not just building some active learning method for image segmentation, but what makes this project special is that we want to retain the out-of-the-box functionality of NN unit, which means that the new active learning components also need to feature an automated configuration based on task fingerprinting. So this ambitious goal taps upon two fundamental open questions in machine learning, which we addressed in two preparatory studies that paved the way for our NN Active project. So since the current literature on the general benefit of active learning is highly inconsistent, we first wanted to know when and how does active learning actually work. So in the first study, which was published at NeurIPS last year, we looked at the current state of the active learning community, which we found divided into some studies stating that active learning does not work and rather just use self-supervised or semi-supervised learning, and other studies proposing new methods and reporting performance records. And we relate this inconsistency to 5 key pitfalls of current evaluation practices, as highlighted here along this active learning loop. And these pitfalls include neglecting important design parameters such as the starting budget, query size, and class imbalance, but also subpar benchmarking due to low quality underlying classifiers, and finally, a lack of comparison against alternative training paradigms like self-supervised learning. And as you can see in the table where a tick means that the method avoided the pitfall, all of the related work is subject to several of these pitfalls. And we address this inconsistent state by proposing a systematic evaluation protocol for active learning and performing a large-scale empirical study where we test these existing methods under meaningful conditions. Now, in the interest of time, I spare you the detailed results tables and I jump right to some of the main insights that we derive from our analysis. So I think most importantly, we found that our random query baseline, where we just sample queries randomly without any uncertainty or clustering intelligence, this random baseline outperforms most methods that are published in literature. That's kind of concerning, right? So if we and that's because we simply ensure that the classifier that's underlying under this active learning algorithms has a good configuration, like a good learning rate and so on. And to address this issue, we propose a lightweight tuning protocol where people can easily tune their classifiers and restore active learning performance. We then find that active learning generally works in class imbalance settings and that the batch method performs best across all settings. So that's one query method for active learning in literature. And we also find that combining active learning with self-supervised learning works really well in terms of performance and other synergistic effects like training time. On the other hand, combining it with semi-supervised learning did not work well. Right. Now, the second question we need to answer on our way to NN Active is how to get useful uncertainty estimates out of a segmentation method. And here again, we find a scattered literature landscape and address it with the systematic benchmarking study, which was presented at ICLR this year by my students Kim and Carsten. And in the first part of our framework, we create a controlled environment allowing to study a potential separation of aleatoric versus epistemic uncertainty. So data-related versus model-related uncertainty. And such separation is in theory desirable for tasks like active learning, where we want to identify samples with high epistemic uncertainty, right? But we find that the question if such separation is actually possible and also if it's really useful in practice is currently unanswered. It's just assumed in current studies. So for instance, there are 2 highly cited studies in the field that are based on the assumption that test time augmentations model aleatoric uncertainty. And if you think about it, this assumption does not really make sense because rotating a noisy image will not add new information about how much noise there is in the original image. However, the fact that the assumption is wrong goes unnoticed in these studies, and instead the assumption is propagated further to subsequent work. And this shows that to study the actual behavior of uncertainty methods, we need a systematic evaluation environment where we can control both aleatoric and epistemic factors. And that's what our framework here provides. Now, the second part of our framework focuses on evaluating the individual components of an uncertainty method to avoid wrong credit assignment. So this includes the often overseen aggregation from pixel-wise uncertainty scores to image-level scores. And the third part focuses on testing existing methods on a variety of 5 downstream applications. So here we argue that uncertainty estimation should be no self-purpose, right? Our observation is that uncertainty methods often miss the link to a clear downstream purpose, and that results in a gap between theory and practice in the field. Okay, again, I will jump over the detailed results and just state some main insights. So we found that separation of uncertainty types, aleatoric versus epistemic, works in toy settings, but not necessarily on real-world data. However, we found isolating epistemic uncertainty can indeed be helpful for active learning and out-of-distribution detection. Also, we find that ensembling is the overall best performing method, and that test time augmentation in fact models epistemic uncertainty. So this directly contradicts the assumptions behind the 2 studies that I showed before. And in fact, test time augmentation is a strong and lightweight alternative to ensembling in modeling epistemic uncertainty. Okay, equipped with this knowledge now about active learning and uncertainties and segmentation, we're currently building this NN active wrapper around NN unit. And on the left side, we see some first results on the hippocampus segmentation task. So importantly, we observed that our query active learning function outperforms the random selection, so that's promising. And we are currently working on rolling out this method to more data sets for robust validation. Okay, now let's shift gears a bit again and get to the third open question that I motivated, which is really about clinical integration of AI support. So given that liability will remain with humans, the successful adoption of AI really depends on whether a clinician can rely on its predictions. So despite the automation provided by an AI system that I exaggeratedly depict here as a red button system, a human still needs to monitor all predictions for their correctness, right? If we develop an algorithm with 95% accuracy, how do we handle the 5% failure cases in life-critical diagnostics, right? Someone needs to monitor this. But if constant monitoring is required, what is the actual remaining clinical benefit of this AI method? And one approach to a more effective clinical decision support are so-called deferral systems, where the model outputs a confidence score together with the prediction. So based on a threshold on this confidence, the human can then trust the model in the high confidence predictions and only revise the low confidence ones. And importantly, from a systems perspective, there are now 2 joint tasks here to solve for the model. So one, the classifier may prevent failures in the first place by just featuring a high accuracy. And 2, we can now detect the remaining failures with this confidence scoring function. Now, when starting my own group 3 years ago, I wanted to build such a system. And of course, I had a look at existing methods first. And it turned out that the first stage of the system, the classifier, is well covered. So as you see here, there are various benchmarks testing the classifier in many scenarios, and the community agrees on the same validation protocol, so mostly using accuracy as a metric. And as you can see, one important aspect here is to test the robustness of systems, meaning their performance under dataset shifts or distribution shifts. So this is because shifts are a major failure source for machine learning systems. And so it's a crucial component to integrate when validating failure detection, right? Because it's a crucial failure source. So here we distinguish so-called covariate shifts, where the image label is preserved under the shift, versus new class shifts, where entirely new categories of images occur that the classifier has never seen before, and so it cannot be correct on these cases by definition. So now when we look at the state of research for the second stage, the confidence scoring, then we see an inconsistent and flawed landscape. There are at least 4 different machine learning research fields that state that their goal is to perform confidence scoring to detect classifier failures. So they all state the same goal, but they have historically grown as siloed fields that do not cross-compare their methods, and they also use different validation metrics that are inadequate for this stated purpose. And they also do not test these confidence scoring methods under dataset shifts, like we have it in task 1. So let's look at the example of out-of-distribution detection, a very popular research field in AI. The majority of current studies in this field states preventing failures of a classifier is the main purpose of the proposed methods. So they really address the task of failure detection that we just saw. However, crucially, the way that out-of-distribution methods are evaluated does not reflect this stated purpose. So this diagram shows the desired behavior of a failure detection method. So output a high confidence for correct predictions and low confidence for all failures. And this task is exactly reflected by the failure label. So meaning when we simply binarize the actual classifier success on each sample to evaluate methods, and then we have this binary label failure, no failure, right? And this guides actually the process of failure detection. But if we now look at how out-of-distribution detection methods are actually evaluated, we see a drastic discrepancy where a different outlier label is employed instead of the failure label. And this outlier label only flags cases from new class shifts as presumed failure. So it only distinguishes between outlier and inlier. And most concerning about this is that the outlier label is generally not defined on all the covariate shifts. And we could argue that those more subtle shifts where the image label is preserved are actually the more realistic and more relevant ones, right? So for instance, we would not expect images of house numbers to suddenly show up in a brain tumor detection task, but we would rather expect subtle semantic variations on this target class. And for these, it's entirely unclear if they're outliers or not, so they cannot be evaluated under the current protocol. So the question arises, why do we employ this flawed outlier label at all if it's only an approximation of the true purpose, which is to detect classifier failure? And remember that the target information, this failure label about the classifier, it is available in these settings, right? So in our study, which was presented at ICLR last year, we proposed a new task protocol that unifies all previously separated fields in the second stage under the umbrella of failure detection. And we conducted a large-scale empirical study. So for the first time, we tested all prevalent confidence scoring functions on various data sets and distribution shifts. And again, I will jump straight to the main insights. So most importantly, we found that none of the many proposed confidence scores or uncertainty functions in literature outperform a simple baseline where you take the maximum softmax response of the classifier. Also, that's again very concerning. And also we found that current OD detection methods, they work well in the far OD settings that they're tested on in the papers, like house number showing up in a, I don't know, animal classification task. But their advantage vanishes rapidly when the shifts become more subtle, which I would argue become also more relevant then. And we also found that the metrics we propose that covers both task 1 and task 2 in one score, it's able to clarify previous ambiguities. So we found a nice metric actually that works well for this task. Right. We then extended the study to the biomedical domain, and we found similar results. And here, the ratio of undetected or silent failures of the current failure detection methods is really concerning for these clinical decision-making processes. So we also developed a visualization tool now that lets users analyze where and why these silent failures occur in their dataset. So visualization here includes various distribution shifts and confidence scores and directly links the deferral system's behavior to the failure image. And this was presented at MICCAI last year. Okay, so the final topic that I would like to highlight is around the ongoing discrepancy between research and actual application relevance. And I showed already various examples for this discrepancy, ranging from the COVID detection study to the various validation pitfalls we found in existing machine learning literature. Another important aspect in this context is the selection of adequate evaluation metrics. Because if we think about it, the evaluation metric defines the task, and so it simulates the application performance, right? And so it can be seen as the glue between research and application. So the most common task in biomedical image analysis is semantic segmentation. So it makes up 70% of the competitions hosted online. And a survey also revealed that the DICE score or DICE coefficient is by far the most commonly used metric for this task. So let's look at a typical segmentation example and see how this metric behaves. On the left, we see a reference mask, and next to it, the prediction of two competing models. Now the DICE simply counts pixel overlap, and so it would prefer model 1 in orange here. But model 1 only detects 1 out of 3 objects or structures in the image. And from a clinical perspective, it's often more relevant to detect all structures. For instance, tumor diagnostics are typically based on lesion load or count and not on individual pixels. So in this case, the problem should have rather been defined as an object detection or instant segmentation problem with a corresponding metric. And this shows how employing an inadequate metric can really hamper clinical utility or guide scientific progress in the wrong direction. So 3 years ago, together with my colleagues Lena Meyer-Hein and Annika Reinke, I started an initiative to systematically address the metric selection problem in the biomedical domain. So for this large-scale initiative, we formed a consortium of experts from over 70 international institutions, which we call Metrics Reloaded. And we also had, and now, so 3 years and many workshops and expert groups discussions later, the project resulted in 2 sister publications at Nature Methods. So in the first, we systematically list the common metric-related pitfalls in the field. I just showed one in the slide before, but this study includes 97 different metrics and 37 pitfall categories. And in the second study, we aim to resolve these pitfalls by presenting a consensus on metric recommendations. So as you already maybe guessed, the metric selection framework, so where we want to recommend what's a good metric for your problem, that's a prime use case for our fingerprinting concept again. So the basic idea is users answer a variety of questions about their problem and what they want, and they so manually extract the fingerprint out of their problem, so to say, by answering these questions. And in the second step, they are then automatically guided through a chain of decision trees. So that's sort of the generalizing solutions that operates in the fingerprinting space. And so the output is a set of adequate evaluation metrics, right? And the scope of this framework includes image classification at all scales. So that includes object detection, instant segmentation, and semantic segmentation. And also the framework is domain agnostic within the biomedical field. This is an overview over the recommendation framework. So most panels here represent an underlying decision tree that guides metric selection for a specific metric category. For instance, the first decision tree maps users to their correct problem or task formulation, so object detection, segmentation, and so on. And this is what this looks like, right? And so if we would have used this mapping on the pitfall that I showed before, we would have avoided the pitfall of using the DICE score for a problem that clinically requires to find all the structures. Now, because each of these studies features more than 200 pages, we also designed an interactive web tool that guides users through the framework. So you can just go on this web page and then click through the questions that you answer about your problem. And then sort of on the right-hand side, as you go through the questions, you start collecting metrics in your basket, so to say. And that sort of facilitates the whole process, and you don't need to read the 400 pages of papers. Right, and now building this framework was a bigger effort than we initially expected. So we were really happy about the positive feedback in the end. For example, Rita Struck, the senior editor that was handling the papers, she stated how important she thinks this initiative was. And that's really good to hear after so many years. And here's also a slow motion GIF where we show our relief when the studies were finally published. Right, so in the end, I would like to talk about the ongoing paradigm shift in AI towards foundation models. So they have shown to enable general purpose AI in other domains, right, like language and so on. So does this mean that they have also the solution to all the problems that I have just talked about? And how can we actually leverage foundation models for clinical benefit? You may know this figure. That's from a perspective article that was published in Nature last year, and it envisions how a general purpose AI in healthcare could look like, and includes combining all clinical data modalities and seamlessly communicate predictions and knowledge with users via language interfaces and so on. So if you're not familiar with foundation models, maybe one sentence. The general idea is now to not have a specialized model trained per task, but you train one general model on a lot of data at a large scale, and then you can easily later adapt this model with very few data to a lot of downstream tasks, right? So that's sort of the paradigm. All right, so how does this vision relate to all the current challenges and open questions that I talked about? As for the first challenge of generalizing models across tasks, I think this problem will shift to the adaptation process. So in the future, the question will be, how can I optimally adapt a foundation model to my problem? So I think the task fingerprinting, like in NNUNet, can be used to automatically design generalizing adaptation pipelines. The described bottleneck will also shift from training to the adaptation phase, right? The annotation bottleneck. So in this context, the question will be, which samples out of my large data pool should I label for fine-tuning the foundation model or for in-context learning? And given the promising results we have seen in our active learning analysis, when combining it with self-supervised learning, we may really see a renaissance of active learning in the future here. Also, I think a major challenge will be to train foundation models on complex data like 3D radiological images. So we haven't really seen self-supervised learning to work in this domain, given the extremely low signal-to-noise ratio. So the needle in the haystack problem, right? How do you get self-supervised learning to work with this? So think about a small liver tumor in a whole body MR volume. So we may need to optimize pretext tasks or smart sampling strategies. For instance, there's some interesting recent work from DeepMind using online active learning to sample informative cases during foundation model training. And as for the human barriers to clinical adoption, I think despite the fact that language will facilitate interaction with the model, the need to rigorously test the reliability and failure awareness of models will not change. So this is why we're currently testing the failure detection ability also of vision language models on our benchmark. And by the way, I think in times of foundation models that are trained on internet scale data, it makes even less sense to categorize cases into inliers and outliers. And we should really just talk about failures of the system instead. And finally, I think it will be absolutely crucial to ensure the clinical relevance of foundation models at all development stages with like thoughtful benchmarking. So this includes thinking about what type of tasks we actually want these models to solve. For example, what is a reasonable trade-off between generalist and specialist? I think of this as a spectrum where on the one side, we have the ultimate generalist with one jointly trained set of weights that solves all tasks via zero-shot generalization, right? This is what often looks like it's promised by the current developments. But on the other side of the spectrum are separately trained specialists for each task. And maybe the optimal solution lies somewhere between these two extremes. For instance, we could imagine a hierarchy of models going from generalist to specialist, and that can interact with each other. So related to this question is the question, which type of generalization gap requires which adaptation scheme? For example, which severity of data set shift do we deem realistic for zero or few shot generalization before it makes sense to fine-tune a dedicated specialist? And what are actual use cases where we do not have access to more than a few examples and where we need to rely on the few-shot approach? And the examples in this figure on the right, I made them up, so I don't know what the actual generalization limits are for each scheme. But if we truly want a general purpose AI with one set of weights in the end, we need to somehow cover all larger generalization gaps above in-context learning. We need to cover that somehow in the training. And my point here is that the benchmarks we use in testing these models already reflect the final application scenarios. So we need to think about this now in order to create meaningful benchmarks and guide the development process. So in the past year, I've been involved in designing and launching foundation model initiatives. I thought this may be interesting to you, and maybe similar initiatives are going on in Norway. So for instance, I was involved in writing the concept behind a project call for foundation models within the Helmholtz Association, where we thought out how to effectively design such projects and teams. So Helmholtz is Germany's largest research association, and it spans 18 research centers from different domains. And in this initiative, we're also interested to connect with similar efforts in other European countries. So for instance, we're currently organizing a joint workshop on foundation models with initiatives from Switzerland, UK, and Finland. And would be interesting to know if something similar is going on in Norway. And as part of this initiative, I'm also coordinating a pilot project, which we launched earlier this year with some colleagues and clinical partners, and we call it the Human Radiome Project. And the idea is that in radiological image analysis, we now have generalist models like NNUNet, But as I mentioned before, the bottleneck of training models from scratch on each new task remains. So NN-Unit is not yet a foundation model. And this is where our human radium shall come in. So we are developing a foundation model that maps complex 3D images into a low-dimensional embedding that represents the full information of human radiology, which is why we call it the human radiome in resemblance to the human genome. We plan to train this model on a data set of nearly 5 million 3D volumes, which we currently collect from our clinical partners. And this data set, it exceeds the largest to date 3D training data set by a factor of 40. So it will really unlock a new scale of training. And we also collected a set of 70 downstream tasks and leaderboards where we want to benchmark the effect and benefit of this model. Okay, now you may say, NN-Unit, that sounds good, but aren't CNNs kind of old-fashioned for this purpose? And shouldn't we use a big transformer instead? So we actually just last month benchmarked the performance of current CNNs versus newer architectures like Transformer and Mamba for 3D segmentation tasks. And it turns out that for the current size of benchmarks, at least, CNN still yields state-of-the-art performance. So we found that the claims in literature about superior performance of Transformers or Mamba models were again due to pitfalls in their validation. For instance, the Swin Unit R transformer used additional training data, which was not disclosed in the paper. And the recent U-Mamba model, which made a big fuss on Twitter or X, it used additional residual connections in the encoder, which was not present in the baselines. So we actually found that the Mamba layers themselves had no effect on the performance. And yeah, I think it will be interesting to see if with the new scale of larger data sets, like in our Human Radiome project, maybe we will see the first true advantages of transformers in this domain. Another important design question in this project is how to reliably track process when building such a foundation model. So I asked my friend, he's part of the AlphaFold team at DeepMind, and I asked him what the single most critical success factor was that AlphaFold works so well now. And he said it was the clear internal benchmark that they had with a clear metric that guided the development process. So as a second experiment in our recent paper here that we submitted to MICCAI, we scrutinized the quality of current benchmarks in 3D medical imaging to check which of these data sets can be such a benchmark that guides the method development process in a good direction. And we define the quality of a benchmark by two factors. So one, we expect high variance across methods. So that indicates that there is an actual signal to measure on this data set for methodological differences. And two, we expect a low intra-method variance. So that indicates a low statistical noise on the benchmark. And then we take the ratio of these two factors, and that sort of also allows us to plot the current popular data sets in this two-dimensional plane. And we find that some of them are really in the red area where the measurable signal does not exceed the statistical noise. And we hope that this nice trick can help identify suitable benchmarks to reliable track process for foundation models also. Okay, that's sort of my final slide. And I thought instead of a sort of summary of research, it would be interesting to highlight some strategies or key philosophies that shaped over the years now for me. So first of all, I think it's important, let's start by saying I'm not so much interested in developing a new method that performs on one narrow task and use case, but rather in generalizing solutions and concepts that have the potential for impact at scale. And thinking big for me also means moving away from individual PhD projects with one student and towards larger collaborations and interdisciplinary teams. So I think this is really crucial if academia wants to stay competitive with cooperative research, right? And I think also your initiative in Norway with the visual intelligence is also one step into that direction, right? And I think we really need to be careful to move fast in this direction if we don't want to lose track. Also, in this talk, we have seen numerous examples where methodological claims made in literature did not withstand rigorous testing. So I believe this is because the pace of our field may come with a lack of scientific rigor in experiment design and evaluation. So sort of in every new topic that we started to do research in, we found an inconsistent landscape of methods, claims, and benchmarks. And I think we need a cultural shift towards viewing systematic evaluation as a necessity, especially when it comes to our own work and methods, to really ensure the meaningful progress. And we sort of need a shift where we value this validation more than a perceived innovation of novelty of some model architecture, right? That in the end maybe doesn't even bring any benefit. Right. Also, in my experience, such rigorous evaluation often revealed that simple and pragmatic solutions work best. So this is also where the name NNUNet comes from. NN stands for no new, so it's the no new net. It's just a very simple approach that's well engineered. And I think we should also appreciate the simplicity, even though it's harder to publish than a novel sort of Mamba-based model or fancy uncertainty function. Yeah, and also I talked about bridging the gap, the translation gap, ensuring clinical relevance, but this obviously also translates to other domain research fields outside of healthcare. And this might be an obvious point, but I think it's really important for this to involve clinicians or domain scientists at all stages of this process to build meaningful methods. Yeah, and of course, let's not forget to have a lot of fun. Right. Of course, none of the work I showed would have been possible without the great team. So this includes, of course, my own group here in the top row, but also my colleagues from DKFZ and Helmholtz, and last not least, the clinical partners in the bottom row. And with this, I thank you very much for your attention, and I'm happy to answer some questions. Thank you.